I mean, we've known for years that bone marrow fibrosis doesn't arise from a single cell type acting alone, but how they cooperate physically, spatially, and molecularly has remained quite elusive over the last few years. And so we previously identified PF4 positive cells, which are mainly megakaryocytes in the bone marrow, as kind of always being in very close proximity to stromal cells. And this is a key population that promotes fibrosis development in MPN. But how is the bone marrow rewired, and how do we interrogate physical interactions in MPN kind of remained a big question. So we now used double genetic fate tracing approach, as well as devised a new bioinformatics pipeline to really interrogate this and really understand how these cells kind of communicate with each other and how this drives disease and whether this kind of changes over time. And so from a kind of more patient perspective, this is quite important, because understanding the kind of earlier stages of the disease and how the different cell populations in the marrow can then kind of be triggered and work together to drive disease basically allows us to find new targets and potentially then new therapies. And so we identified SPP1 positive macrophages as being a really important cell population in this context. And we see that SPP1 signaling is something that happens in bone marrow fibrosis. And so being able to maybe manage that, or maybe manage the amount of macrophages that you have in the marrow will be important going forward.